Okay, let's take a look at a non-resistant cell first, and say we treat it with calcium AM, and the calcium AM goes inside the cell. Now, the influx is going to be much faster than the efflux just because of the concentration gradient, because you've got more outside than inside. Okay, inside the cell, the calcium AM is hydrolyzed to calcine, which is polar and fluorescent and can't get out of the cell readily. So very little calcium AM actually comes out. Now these cells with calcium trapped inside are now highly fluorescent, and you can measure the fluorescence of the cells directly. Okay, let's take a look at an MBR1 cell which has pump activity. Now if we administer 60 nanomolar calcium AM, the pump is so fast that it gets the calcium AM out of the cell before hydrolysis can occur, and these cells are completely non-fluorescent. Okay, so in this situation where you've got a resistant cell that has pump activity, you've got 100% efflux, you have zero retention of calcium, and the calcium AM just goes right through the cell, and you don't see anything in your fluorescence instrument. For the latecomer, I'll just mention that this is a non-resistant cell, and the calcium AM goes in much faster than it goes out. And it's hydrolyzed by the esterases inside the cell to give free calcium, which is polar and highly fluorescent. The calcium AM is nonpolar and not fluorescent. Well, are you talking about non-resistant or resistant? In a resistant cell, it could be that over time you'll start to see some fluorescence. I don't know. I mean, I've never followed it for longer than about. No. That pump is very powerful and it's very fast, and that's why it's a major survival mechanism in biology throughout nature. You'll find it in bacteria, you'll find it in Leishmania, you'll find it all over the place. Okay, now let's take a look at the resistant cell again. We administer the calcium AM. Now we're going to preload the cell with 9.5 micromolar PSC 833, which is reputed to be the best inhibitor known so far. And I'll show you in a minute how we got to that concentration. This concentration of PSC completely blocks the pump. Now the calcium AM goes into the cell. It's hydrolyzed to free calcium, which is trapped and fluorescent. So you get, in this situation, you get the maximal fluorescence that you can get with the system. So now we have zero efflux and 100% retention rate. So actually when we do these experiments, we have two standards. One is the control, where you have the fastest efflux and zero retention, and the other is the fully inhibited sample, where you have 100% retention and zero efflux. Well, what happens when you partially inhibit the pump? Let's say you go to a concentration that's about in the middle. Well, 1 micromole of PSC 833 will be about at the middle. The calcium AM will go in. Part of it will be hydrolyzed and trapped, and part of it will be released. Now, if you take the percent of the retention rate plus the percent of efflux, you get 100%, because there are two pathways that the calcium AM can go through, and there isn't any other major pathway. And this is the only original idea in the whole study, was namely that if you add up retention and efflux, it's going to be 100. So the first thing to do, you've got to convert all your data to percent. Now, for those of you who do tissue culture, will know that no two samples on different days are ever the same. The amount of pump may change, the viability of the cell may change, the metabolism of the cell may change, so that you always have to have standards for each day. And the standards in this set of experiments is the control, which is 0 and 100%. Okay, this instrument doesn't exist in this form anymore. There's an arc lamp in the back, single grating monochromator. The sample is here, and then it goes through another single grating monochromator, cooled photomultiplier tube. The data is stored in this box, and then the stored data is transferred to a computer. This is very old. This was a 1982 model. This water bath was set at 37 degrees, and the cell suspension was placed in here just before the experiment was run. And then the water was pumped through a chemical pump, which you can see peeking out of the back, and I put some insulation on the tubing to try to hold the temperature. By the time it got into the cell compartment was 35. So we've lost 2 degrees, and I couldn't see any way to get around that because these cells don't like to be too hot. If you get up to 38 or 39 degrees, they're going to get sick. So the whole goal of the experiment is to try to keep the cells alive throughout the whole experiment. The data actually comes out on an XR reporter over here, and I'll show you lots of data. Okay, the reaction is run in a plastic cuvette with a little stir fin, and there was some magnetic stirrer built into the bottom of the cell compartment, which also worked on the rotoflow. And I put that in myself. I killed a whole month just to get that damn thing to work. Okay, now this is an experiment with non-resistant